Hi, and welcome to Metro News 1. I'm your host, Ella Sanders. Today, we have some guests coming to chat with us about a genetic technology called CRISPR Cas9 and how it's being used to treat the Duchenne Muscular Dystrophy gene in animal models. Our guests are experts in this field. First off, we have Thomas Mantouffet. Now, Thomas, can you explain to me what is CRISPR Cas9?
Thanks, Ella. Happy to be here. So, CRISPR Cas9, now bear with me while I say this, stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a genetic technique in which molecular biologists are able to modify genes within a living organism. More specifically, we're interested in its ability for human DNA. Its potential is largely used from discovering new medicines, genetically modified plants, also to the intervention of genetic diseases. So the way CRISPR works is by introducing this Cas9 protein to the target DNA strand along with a gRNA, known as its guide RNA, containing the new set of genetic material, splitting the DNA into RNA, and thus allows the scientist to replace the pre-existing genetic material with the new genetic set. This is very vital for today's day and age where common diseases are more genetic based rather than bacterial or virus. So this change, for example, can stop the expression of genetic disorders such as Duchenne Muscular Dystrophy.
Wow, that's amazing. Just another quick question for you, Thomas. What exactly is Duchenne Muscular Dystrophy?
Now, Duchenne Muscular Dystrophy, also known as DMD, is a severe muscular disorder that affects mostly boys, approximately 1 in 4,000 to 5,000 live births, as well as, but more rarely diagnosed in girls, about 1 in 50 million in live births. An early sign is muscle weakness occurring around 4 to 5 years of age and becoming more severe towards that 10 to 12 years of age, where it is almost impossible to walk. It is known that this disorder is directly linked to the X chromosome in the recessive gene, where it is inherited 67% of the time, or in the other 33% of the time, from mutation within the body. This genetic disorder causes the improper development of the dystrophin protein required for different muscles. A quick fact that the gene that codes for dystrophin is the largest human gene known in the DNA sequence. And the use of dystrophin in the body is vital for movement, such as your skeletal muscles, and for the circulation of blood, or the main pump your heart, your cardiac muscle. Now, unfortunately, there is no current cure for Duchenne muscular dystrophy. However, treatment is more targeted towards the minimization and the controlling of symptoms rather than actual cure. This is to provide a better quality of life for our patients.
Wow, thank you so much for coming on the show. Next up, we have Tyler Lang. Tyler, tell us, how is CRISPR used in animal models for Duchenne muscular dystrophy?
Hey Ella, my paper looks at using CRISPR Cas9 to stimulate dystrophin expression in canine models. The Delta E50MD dog model, sourced from dogs which naturally present the disease, provides an accurate analog for the human DMD gene. Adeno-associated viruses were used to deliver CRISPR gene editing components to 4 dogs. The dystrophin levels monitored over a period of 6 weeks resulted in an 80% recovery of wild-type levels. Thousands of mutations have been associated with the dystrophin gene, which contains 79 exons. Most mutations cluster around a particular area ranging from exons 45 to 50, placing exon 51 out of frame, with preceding exons preventing the expression of functional dystrophin protein. Modern treatments involve an oligonucleotide, which is a short DNA or RNA molecule, to skip exon 51. This results in dystrophin expression of 0.22 to 0.32% of normal levels after 1 year of successful treatment. However, it is estimated that only a 15% restoration would provide significant health benefits. Prior to the experiment, CRISPR-Cas9 had been used to restore dystrophin expression in mice and human muscle cells derived from human stem cells. Adapting the technique in vivo in a large mammal further validated CRISPR-Cas9 as a viable treatment for DMD. To correct the dystrophin reading frame in the canine model, Streptococcus pyogenes Cas9 coupled with guide RNA targeted a region adjacent to exon 51. For in vivo delivery of Cas9 and the guide RNA to skeletal muscle and heart tissue in dogs, a recombinant adeno-associated virus, serotype 9, or AAV9, was used. AAV9 displays a high selectivity for skeletal muscle and heart tissue, a muscle-specific creatine kinase, which is an enzyme which catalyzes the conversion of creatine, was used to drive Cas9 expression, and 3 RNA polymerase III promoters, which transcribe DNA to form short RNA sequences directed to the expression of the guide RNA. 2 doses were tested, 2 times 10 to the 13 vector genomes per kilogram and 1 times 10 to the 14 vector genomes per kilogram. The delivery of the larger dose, 1 times 10 to 14 vector genomes per kilogram, resulted in widespread dystrophin expression after the experimental period to a markedly greater extent than the lower dose. This adjacent image of an immunohistochemical stain of various muscle samples with a healthy model on the right, an untreated model second from the right, the lower dose second from the left, and the higher dose on the left demonstrates the widespread expression of dystrophin across multiple muscle types. Thank you.
Isn't that just incredible? Thank you so much, Tylo. Last but not least, we have Anthony Feek joining us. There were some technical difficulties for Anthony, but not to fret. We are still able to chat with him about the use of animal models for the CRISPR Cas9 gene editing on the Duchenne Muscular Dystrophy gene. Take it away, Anthony.
Yeah, so due to the nature of such experiments, there is great potential that test subjects may face diminished lifespans and/or face serious pathological issues. It is therefore more desirable to use animal models in an ethical way as to not endanger a human's life or quality of life. Animal models also happen to be considerably cheaper than human models, allowing laboratories to conduct more thorough and extensive investigations within a tight budget. It should also be noted that many animal models can display pathological conditions and have those conditions progress at rates considerably faster than in humans, furthering the argument as to the usefulness of animal models. So actually, although animal models do possess a multitude of advantages over human models, there are several issues that need to be considered. Firstly, animal models are very prone to suffer from unethical treatment and often die painfully. Hence, many ethical concerns arise from the use of animal models. Another important issue is that animals do not precisely reflect humans in how their bodies react to certain diseases. Therefore, all the data achieved using animal models cannot be said to be fully accurate and can only be applied to humans with a degree of error. Rabbits reproduce. They are a preferential candidate for CRISPR-based investigations. Rabbits are also extremely easy to look after, and expensive equipment is not needed to look after them properly. A drawback of rabbit models is that the pathological responses and general physiology of a rabbit differs rather significantly from humans, meaning the experimental data cannot be accurately applied to humans.
Thank you so much, Anthony, for all that information. That's all for today. We hope you've learned a thing or two about the use of CRISPR Cas9 for treating the Duchenne Muscular Dystrophy gene. I'm Ella Sanders, and this is Metro News 1.